Hey guys, this presentation is going to be about bacteriophages, but more specifically, how the experiment pertaining bacteriophages done by Alfred Hershey and Martha Chase led to discovering that DNA is a carrier of genetic information. Now, wait. Isn't it obvious that DNA is what carries genetic information? I mean, that's what we knew all along, and this is what we've been learning since middle school, right? Well, not exactly, at least not during the early 1950s. At this time, proteins were considered the carrier of genetic information. Previous experiments known as the Avery McLeod-McCarthy experiments displayed results supporting the idea that DNA, not proteins, carry genetic information. This idea was heavily criticized due to concerns towards the validity of these experiments and, in turn, sparked Hershey's interest. Wanting to settle this criticism towards what was recognized as a rather well-supported and endorsed set of experiments, Dr. Hershey conducted his own experiment. Now, before we get into detail about this experiment, a couple of things should be addressed. Dr. Hershey was well known for this experiment and ended up receiving a Nobel Prize for his works. Wait, his works? Martha Chase definitely contributed to this experiment as an assistant, but from what information can be gathered of her, she didn't get nearly as much credit, nor is she described as being more than just an assistant. This includes her motives or what she actually did in the experiment. Even so, her works and efforts contributed greatly towards a big step forward in understanding DNA and genetics as a whole, and at the very minimum will be known to by our class. Going into the experiment itself, bacteriophages, well, bacteriophages definition. Bacteriophages are defined as a virus that parasites a bacterium by infecting it and reproducing inside it. Yeah, I'm just gonna let's copy that link. These phages reproduce by penetrating the bacteria cell and placing in genetic information into the bacteria cell, in which bacteriophages would then reproduce inside the cell. And then the cycle continues. This was more or less the understanding of bacteriophages during this time as well, aside from specific parts of the virus and its structures. But what was uncertain was what exactly was holding the genetic information and how was it being passed through. The idea that proteins held this genetic information was most widely accepted due to scientists observing 20 different amino acids being present for the creation of a protein polymer, in comparison to DNA polymers being made of only 4 nucleotide bases. For the experiment, bacteriophages were placed on the bacteria pellet, which contained bacteria cells. Half of the phages placed on there had their proteins marked radioactively, and the other half didn't. It was left unmarked. There was another block or group in which half of the phage's DNA placed on the bacteria pellet had their DNA marked radioactively, while the other half was, again, left unmarked. Each group was placed in their own blender, in which the phages and the bacteria cells were blended until the cells and the phages separated as a result of centrifugal force. Blending the cells and the phages resulted in a revelation that most of the radioactive proteins were found in the capsid head, or the phages themselves, whereas most of the radioactively marked DNA was found in the bacteria cell. Along with multiple trials and further repetition, well, take it away, Sam.
This demonstrated that DNA, not protein, provided the genetic material that helped define the growth, development, functions, and reproduction of organisms. And we can see this during the experiment because DNA was transferred to the bacteria. During the experiment, the bacteria pellet was radioactive when infected by the radioactive phosphorus, but not when it was exposed to sulfur. The DNA component of the bacteriophages is injected into the bacterial cell that is able to direct the formation of new virus particles complete with protein coats, while the protein component remains outside. These results were discovered from the discovery that DNA, not protein, was transferred to bacteria. This was revolutionary in helping convince the world that DNA was the genetic material. It led to biology's next important objective in leading to the research of the structure of DNA. This is reflected by the findings of the double helix by Francis Crick that only came 11 months after the Hershey experiments.